So our actor today is liver fluke, is Fasciola hepatica, and you can see on this review in Trends in Parasitology that it is a very important parasite, as we know. And in this paper of last October, it is considered the parasite of the month. So there are still many challenges regarding the epidemiology, regarding diagnosis, and regarding treatment. And today we will focus on diagnosis, and you can see, and during prepare, we have agreed on the fact that diagnosis can be achieved primarily through the microscopic detection of the eggs of Fasciola hepatica using sedimentation or flotation techniques, as we will show today. But also immunodiagnostic is important, and also molecular tools. It's important to look at coproantigens in the feces or other antigens in the blood, in the serum, and even in the milk of infected animals. Here in this paper, which is a practical guide to the diagnostics of ruminant parasites, including nematodes, liver fluke, and lungworm, you can see the availability of different text, when to use it, the sampling, the diagnostic value, and also the drawback. So as I said, we can use blood antibody ELISA from 3-4 weeks post-infection, coproantigen ELISA from approximately 8-10 weeks post-infection, milk ELISA, which is very useful in dairy cattle from 2 to 4 weeks post-infection, or we will focus on copromicroscopy, so the egg counts of this parasite from approximately 10 to 11 weeks post-infection, and this depends on the biology of the parasite, and in particular on the prepatent period of the parasite. So you can see also the different diagnostic value. And copromicroscopy is important because it allows, as the post-mortem diagnosis, to perform a final diagnosis because eggs will be produced only when adult parasites are present. But there are a lot of challenges related to copromicroscopy, as we will discuss later on. I thank Lavinia for these accurate slides. Again, you can see the sensitivity and specificity of liver necroscopy, so post-mortem examination. So specificity, of course, is 100% because the adult parasites are well known and can be easily seen. But sensitivity can be 93% because you have to do accurate cuts of the liver. By examination, 81% sensitivity and 97% specificity. And regarding fecal egg count, sedimentation has 100% specificity because you can detect the entire eggs, but sensitivity is 68%. Fluke Finder is a very good tool to perform fecal egg count with 80% sensitivity and 100% specificity. And today we will talk also about mini FLOTAC, because also with the flotation using zinc sulfate as a flotation solution, we can reach high percentage of sensitivity and specificity. So fecal egg count as a post-mortem examination is a direct diagnosis, whereas we can also use some methods for indirect diagnosis as immunological techniques, so antibody detection in serum, antibody detection in milk, or antigen detection in feces. And on adult parasites or on the eggs, we can also use molecular techniques, which is a direct diagnosis, so PCR, RT-PCR, PCR-LFP, or lab. So there is a wide portfolio of diagnostic tests, but today we will focus on copromicroscopy. Okay, so Maria Paola is here, and she will present the copromicroscopic diagnosis of Fasciola and Paramphistomidae in cattle.
Okay, for a good treatment, it's very important that we perform a good practice of a diagnosis. And even if we are in the era of genomics, metagenomics, proteomics, and bioinformatics, diagnosis of endoparasites largely relies on copromicroscopic techniques. The microscopic coprology started in 1857. And science since this year, today, many different veterinary and human techniques, copromicroscopic techniques, have been developed. And you can see the difference for human technique with the red line, the veterinary techniques with the blue lines, and also different commercial kits have been developed with indicated with the black lines. For a good diagnosis, it's very important to perform a good practice also for sampling, preservation of the samples, and the preparation of the samples. Regarding sampling, we suggest you to collect individual rectal fecal samples from 20 animals, from 20 cattle in each farm. Weaned animals at first grazing season are preferable, and for the transport of samples, we suggest you to use a temperature don't below 10 degrees. We have performed different studies on preservation of the samples. These are the results of our study of a comparison between fresh formalin 5%, 10%, and frozen fecal samples. We have used this comparison for the diagnosis of gastrointestinal nematodes in a sheep. And you can see that with the red lines, we indicate the mean number derived from 6 replicates of eggs per gram of feces, and with the blue line, the coefficient of variation, that is a indication of the precision of the technique, and is calculated using the standard deviation divided the mean EPG per 100%. And it's very important that the coefficient of variation is very low to have a precise technique. And you can see that with the frozen preservation, we had the worst results. And so we have also compared the fresh feces with the vacuum packaging. And in this study, we have compared feces without vacuum at fridge temperature, vacuum packaging at plus 4 degrees, and the vacuum packaging at room temperature. And you can see that we have performed these assays for 28 days, and up to 21 we obtained the good results with the vacuum packaging, packing at the fridge temperature. Also, we have performed different studies on preparation of samples. We have performed a study using individual fecal samples, but also pool samples with 5, 10, and 20 samples. And we obtained good results, a good correlation between individual samples and the pool samples. In particular, if you see the first in this part, this graph is for the correlation between individual samples and the pools of 5, and the correlation is very optimal. And regarding the copromicroscopic techniques that you can use for the diagnosis of Fasciola and Calicoforon, as Laura mentioned before, you can use flux finder, sedimentation, and the mini FLOTAC. And I remember you that the standard operating procedures and the video, but also in Atlas with the parasites that you can find with these techniques, it's available on Prepare website. We have compared these three techniques for the diagnosis of Fasciola and Calicoforon dabni in cattle. In particular, we have used the egg-spiked fecal samples with the three levels of infection, 10, 50, and 100 eggs per gram of feces. And we have used also natural infected fecal samples, 10 cattle with the 20 cattle from 10 cattle farms. And you can see also in this slide the schematic features of mini FLOTAC, finder, and sedimentation. And so you can see the amount of feces used for each technique, the quantity of feces examined, and the detection limit and the multiplication factor used. You can see that for the level of 50 pg and 100 pg, we obtained the higher egg recovery percentages for mini FLOTAC. And for the lower level of 10 pg, we obtained the best results with the mini FLOTAC and the finder, and the worst with the sedimentation. We have also compared with the feces naturally infected. We have compared the individual fecal samples and the pooled fecal samples. And you can see that the mean, the EPG mean, it's very similar between the mini FLOTAC, the pooled samples with the mini FLOTAC, and the individual fecal samples. Instead, the results obtained with the flux finder and the sedimentation are not very near the true value of EPG. And for the mini FLOTAC technique, you know that the steps are 5. In the first, you add the flotation solution, and in particular for Calicoforon and for Fasciola, we use zinc sulfate, as Laura mentioned before. And in the second step, we add 5 grams of feces, and so we use a dilution factor of 1 to 10. We homogenize the sample, and in the fourth step, we filter, and contemporaneously we fill the 2 flotation chambers, wait 10 minutes, translate, and examine the mini FLOTAC under the microscope. In this case, the analytical sensitivity and multiplication factor of the technique is 5 eggs per gram of feces. And later we will see all the steps with Lavinia, with Antonio. And it's very important to mention that with the flotation, you see only the egg shells of Fasciola that is indicated with the red arrow, and the Paramphistomidae that is indicated with the blue arrow. But later we will see each step in details. Thank you for attention.
After the presentation of Maria Paola on mini FLOTAC, which is a flotation-based technique, we will share with you our experience on the use of artificial intelligence in parasitology. I think it's very important when dealing with the parasites, and in particular with liver and rumen flux, because you can see the different topics that can be addressed using artificial intelligence. For example, for epidemiology and disease forecasting, because artificial intelligence can combine climate sensing, epidemiological history, and we know that rumen and the liver flux are very dependent on climate and environment. So using AI, we can handle big data, we can perform early warning systems or aid in better preparedness. It can be used also for drug discovery in order to find new target identification, novel drug, and drug repurposing, because there are challenges for liver and rumen flux, not only regarding diagnosis, but also regarding treatment. So we hope that in the future, a vaccine will be available for liver flux. Today, we will focus on the application of AI in diagnostics. So using machine learning and deep learning algorithms to train a large data set in microscope image analysis. And this can allow higher sensitivity and specificity, the use of smartphone application and implementation at resource poor setting. So in the end, AI can optimize energy consumption and improve resources management by combining and coordinating expertise from different domains. We are in the era of technological innovation and digital transition. So really, the technology is advancing a lot in order to use smartphone-assisted diagnosis to artificial intelligence. And in particular, we will focus on automated systems for parasite count based on AI. Some years ago, in 2022, we have published this chapter in Advances in Parasitology in order to show the advances in diagnosis of gastrointestinal nematodes and other parasites in livestock and companion animals. So nowadays, there are different systems for parasite counts based on artificial intelligence. But I would like to underline that the human expertise, so the knowledge and the expertise of parasitologists are very important, because supervised learning depends on parasitologists, because the first step is to label parasitic stages, and then we have to extract the deep learning models. So it's important to not neglect human expert knowledge, because otherwise even a very power AI system can be not useful. And it's important the integration of knowledge from parasitologists, which plays a pivotal role in refining the performance of deep learning models. Here you can see from our review how many methods are based on semi-automated or automated systems for parasite detection. And here you can see the name of the method, the principle, the host which apply the parasites, but also a SWOT analysis with strengths, weaknesses, opportunity, and threats. There are some that have been already used for diagnostics of parasites, as the KFM we will talk about in a while, the parasite system, but also the vet scan images, which is very much used in veterinary field, or also the lab on disk platform. And also in human parasitology, there are different methods that can be used using an iPhone, for example, or the FACPAC I have talked about, or other systems that can be used to detect parasitic elements in stool or in urine samples. It's important to consider that some methods have reached a TRL, a technology readiness level of 4 and 5. So they are at the stage of simulated world. So they are at the phase of prototype. For example, this lab-on-disk platform, the automated diagnosis of intestinal parasites DAPI, the Kubic FLOTAC microscope, and the digital diagnostic system for soil transmitted helminths. But there are also other that have reached a TRL, a technology readiness level of 8, 9. So they are already in the production phase. So some of them are already complete and qualified, and they are on the market. For example, in human parasitology, the Spot Lab or the enablers, and in veterinary parasitology, the microkit fecal egg count, the FECPAC, the vet scan images, or the telenostic. So again, some are at the prototype level, some other are already on the market. Of course, to develop a system based on artificial intelligence in parasitology is very challenging because we should use expertise from veterinary and medical parasitology, but also expertise from engineering, electronic engineering, mechanical engineering, optical engineering, and IT engineering. And we started using these automated systems, then linked to artificial intelligence already more than 10 years ago. We started with some prototypes and with the reading of the slide on a smartphone. So here you can see that already in 2012, there was the development of the first prototype of the mini FLOTAC automated system. Then we started collaborating with engineers. Then there was a first validation of the system as a digital microscope for fecal egg count. And 4 years ago, we started a formal collaboration with the Department of Engineering in order to implement a deep learning and an AI and model training for selected parasites of animals. And recently, we within the Prepare project, we trained the system for liver fluke and rumen fluke. As I said, the Kubic FLOTAC microscope is at prototype level. So the TRL is between 4 and 5. And nowadays we have 20 prototypes that are handcrafted. And you will see one of these prototypes working in our lab. Of course, it's really demanding. There is a lot of work, but also a lot of economic commitments related to the development of this microscope. Here you can see, for example, the different equipment, so the digital camera, the LED light sources with the condenser, but also the motorized stage to adapt the mini FLOTAC. So really it's a very, very demanding activity. As I said, we started the first trials and the first validation for gastrointestinal nematodes in cattle, and we found a very high concordance correlation coefficient between the optical microscope and the KFM. So the system has been already validated for gastrointestinal nematode in cattle, and in this study we performed this trial at the Ghent University in Belgium by spiking the fecal samples of cattle with the different levels of EPG, so low level, medium level, at high level. And here you can see that the correlation was very high. And thereafter, we tried to improve the system in the following years, and in particular to implement the the deep learning and the artificial intelligence software. Again, on gastrointestinal nematodes, this is Paola with Candela. We did these trials in Italy and in Argentina, and we found a very high average precision when performing KFM and artificial intelligence for gastrointestinal nematodes of cattle. Another manuscript is in preparation within the activities of Prepare, because we trained the software for Fasciola hepatica and Chalicoforum daubni, so the liver and rumen fluke of cattle, and we found a high mean average precision. This value is an important value because it incorporates the trade-off between precision and recall, and considers both false positive and false negative. And it's very important also to consider the quality of the fecal sample, so the presence or not of debris. Here you can see some eggshell of liver fluke and rumen fluke at the microscope, and you can see that when the clarity of the sample is better, as in this picture, there is a higher accuracy because there are the absence of debris or the low presence of debris. So the post-processing detection will have to fix some imperfection and increase the average precision value even for more concentrate samples. We will show in a while the KFM automated scanning steps. First there is the mini FLOTAC preparation, as Maria Paola showed. Then there is a scan session, actually is 6 minutes. Then there is the image delivery in the server, and a chamber of the mini FLOTAC contains 192 images. And then after 8 minutes, there is a report generation. So the system of artificial intelligence can detect, as in this case, 0 EPG of Chalicoforum, 10 EPG of Fasciola, and 56 of gastrointestinal nematodes. And here you can see that, but Giuseppe will show you later on, that the first step is to train the software. So here you can see that the first step is labeling. So we had to label the air bubbles and the parasitic elements, in that case Fasciola and Paramphistomidae eggs. So there is a 70% of training session, 50% for validation, and 15% for testing. We have done this trial on the efficiency of the artificial intelligence system for Paramphistomidae and Fasciola hepatica in different animal species, so cattle, goats, and sheep. And here you can see how many labels we have done in the lab. So the system has been trained with more than 12,000 Fasciola eggs and more than 20,000 Chalicoforum eggs. And here you can see that these eggs, based on the artificial intelligence system, can be distinguished from other parasites, but also from pseudoparasites. So really it's promising for ruminants and humans. Here you can see the workflow. So the labeling and the scan images collection, the images pre-processing and color grading, the deep learning eggshell detection, the overlap aware and knowledge-based filtering, and finally the report generation. And we have done lots of internal test set and external test set because we aim to try and to compare the optical microscope and the AI system on 100 fecal samples from cattle. So the first step, as I said, is to label the eggs, to train the artificial intelligence system, and then the validation is based on the comparison of the EPG between an optical microscope with a technician and a reader and the KFM scan. Okay. Of course, there are different strengths, weaknesses, and we have future perspective. Among the strengths, it is a portable microscope, and ideally it can be used also in the field. It allows automatic scanning, multiple egg class availability, can have a user-friendly web interface and an app. We can use in remote. As I said, the battery, it's based on lithium and can work up to 20 hours. And hopefully it will be low cost. But we still have some weaknesses. For example, some mechanical issues, optical problems, autofocus should be improved, and also we have to insert a battery icon. So the future perspectives are to reduce the time of automated diagnosis, training the neural network also for other parasitic species, and having a recognition of parasite without internet access. So we are already working with a microscope company because, as I said, the recognizing of the eggs by artificial intelligence is done. The efficiency is very high, the correlation is very high, but we have to implement the system by reducing the scan time from 6 minutes to 30 seconds per chamber to improve the quality of the images obtained, but also to improve the mechanical parts and battery performance.